All right, I think we're going to go ahead and get started. So welcome everyone, and thanks so much for joining us today. First off, my name is Willie Goldsmith, and I'm a senior associate with the LenFest Ocean program. And for those of you that don't know us, LenFest is a grant-making program that funds research projects and expert working groups to address priority science needs facing ocean and coastal decision makers. And if you'd like to learn more about LenFest, feel free to visit our website at lenfestocean.org. Where you can also sign up for our newsletter. And we also encourage you to follow us on Twitter, where our handle is @LenFestOcean. So for today's webinar, we're excited to have with us Drs. Gavin Naylor, Shannon Corrigan, and John Denton, who are all from the University of Florida. And they'll be providing us with an update on their application of genomic methods to investigate the population structure of the Thorny skate in the North Atlantic Ocean, and also to explore factors that may have contributed to past changes in abundance for the species. And we here at LenFest are really excited about this work and the potential for these genomic tools to inform the conservation and management of species like the thorny skate, as well as others for which key stock structure questions remain unclear but are really needed to provide sound management guidance. So before I turn it over to Gavin, Shannon, and John, I just want to go over a few quick logistical items. As you may have noted, we have all attendees muted just because we have a bunch of people on the line and want to prevent any feedback or echoes from coming through. After the research team provide their presentation, we'll have the last 20 or 25 minutes available for questions. And again, because we have folks muted, we encourage you to submit your questions using the Q&A panel in WebEx, where you can type and submit your questions. And we'll be keeping track of the queue there, and I'll read it aloud at the end for the researchers to answer. And depending on how many questions we get, we might not get to them all, but folks are certainly welcome to follow up with us or with the researchers, and at the end we'll have their contact information available. For next steps, we know that not everybody's able to make the time today, so we will be recording this webinar, and in the next couple of days or a week or so, we'll be making that webinar recording with a video available on our YouTube channel, and we'll also be distributing the link by email. I know some of you are on the running distribution list that I have for this work, but if you're not on that list or you're not sure if you are, just shoot me a note. My email address, as you can see, is on the screen you see right now. And we'll also be live tweeting today's webinar if you'd like to engage with us there using the hashtag #NaylorWebinar. So please feel free to use that if you would like. So with that, I'm going to turn it over to Gavin, Shannon, and John, and I'm going to pass presenter privileges over to you guys now. All right. Okay, you guys should be good to go.
1 second. Okay, how does that look?
Looks great.
Perfect.
Great. Thanks very much, Willie. This is Gavin Naylor here. We're excited to share the progress that we've made so far on this LenFest-funded project. But the first thing I'd like to outline is that the project is not just for University of Florida researchers. We are genomicists and population geneticists and phylogeneticists, but the thorny skate problem was first introduced to us actually by Dr. Jeff Kneebone at the New England Aquarium. He's really the lead on the idea for this particular project. He told us, he's been studying thorny skates for a long time, life histories and their movements through tagging, and he told us some of these bizarre patterns that defied explanation, and we became interested in them and wanted to apply genomic methods to see if we could shed light on what might be going on. Similarly, we're not the only ones to have thought of doing this. Dr. Arve Lingerhammer at the Outtakes University in Norway has done some initial work using molecular data to try to understand the conundrum that the thorny skates present. And he's been an absolute prince. He's shared all of his data and collaborates with us, sent all of his tissue samples over, and is very keen to try to get to the bottom of these problems. And then also, because we're taking a genomic approach, we need a high-resolution genome so that we can be sure that the comparisons that we make are meaningful. So we've teamed up with the Vertebrate Genome Project at Rockefeller University, specifically with Dr. Olivier Fedrigo, who's been great and has been working to try to put together a high-quality chromosomal level assembly for the Thorny-scaped genome that we can use for this project. So we've also been helped by a lot of fisheries biologists, particularly Dave McElroy at the Northeast Fisheries Science Center, Dr. Clara Jakobsdottir over in Iceland, and Mark Simpson at Fisheries and Oceans Canada, who have been incredibly selfless and sent us tissue samples for hundreds of these animals from their particular regions. And we wouldn't be able to do any of this work if it weren't for these 6 researchers. The motivation for the problem came from Jeff, and all of them have been extremely helpful in trying to help us get to the bottom of these problems. Now, the 3 of us, myself, Shannon, and John, we've wanted to tackle this in the best possible way, and Shannon and I wrote a proposal to the Lenfest Ocean Foundation, which has been endorsed and funded. And the way we're going to do this presentation, I'm just going to introduce the problem, and then Shannon, who's been instrumental in developing many of the techniques that we're using in gene capture, will then talk about the approach that we're going to use. And then John Denton, who's recently joined our team, has actually been collecting the empirical data and analyzing the data, and he will tell you what we've found at this stage and what we're hoping to look forward to for the next phase of the work. So let's get straight in to the Thorny-scape. So the Thorny-scapes in the genus Amblyraja, and there are 8 of them in the genus, different species found in different parts of the world. And this one at the bottom right-hand corner is the Thorny-scape. This is the animal whose biology we're keen to try to understand. And it's distributed throughout the North Atlantic, from the Gulf of Maine up through Canada, on the western coast of Greenland, the eastern coast of Greenland, completely surrounding Iceland, the UK, up through Norway, and all the way up into the Arctic Ocean and Svalbard. Now, the Thorny-scape overlaps in distribution in New England waters with 6 other scapes, shown here. And we refer to this as the New England Scape Complex, and the New England Fishery Management Council manages all of these scapes sort of collectively. Both they monitor individual species catches, but also manage them as a group. And they've been recording data rigorously since the 60s. And here is a graph where you can actually see landings in metric tons for the scapes collectively. And we can see that it's a very sort of bumpy graph. Some years are good years, some years are bad years. But you can see that extending from the 60s all the way to the year 2000, there's been an increase in the landings of these scapes. But you can also see that there's a dotted line, which is a little bit alarming. This is the survey index of thorny scapes, the biomass. And that, since the peak in the early 70s, has actually been trending downwards to an alarming degree. And the last date here you see on this graph is 2000. Well, this has prompted a management plan to be set up. And this was set up in 2003 to actually manage and protect the thorny scape populations in New England waters. So in this graphic, you can see a bold red line that corresponds to 2003, and we can see 7 different scapes and their population numbers, their biomass index specifically, and how it has been changing over time. And you can see, for example, the barn door scape is recovering very nicely, and the others are sort of bouncing around there's a lot of stochasticity in it. But what is particularly problematic is the thorny scape here is actually not recovering whatsoever. So this is a concern, and this is a concern that has caught a lot of people's attention. What's intriguing is that if we look at the biomass in the northeastern Atlantic, over in Norway and the UK in the North Sea, we see that the numbers are actually increasing, and they seem to be fairly healthy. So it's somewhat intriguing that in the Gulf of Maine, the numbers are diminishing and not recovering, when measures have been put in place to protect them. But in the Northeast Atlantic, shown in this particular graphic, they seem to be doing fine, or at least they're sort of stable. Now, the problem is made more complicated by the fact that in the Gulf of Maine, there appear to be two size morphs that are sympatric. There are some that are late maturing and live longer and attain lengths of about 85 centimeters total length, and they take much longer to mature. And there's a second class that matures earlier. And we know that these are two distinct size classes because we find large individuals which are immature and yet larger than the small form which has already reached maturity. It's almost as if there are two sort of cryptic forms, cryptic species in the population, but otherwise they're indistinguishable. We can't tell the difference except from these life history traits and their time of maturation. Now, the other thing to understand about thorny skates is they don't move very much. In 1984, Templeman reported movements of tagged animals over a 20-year period, and none of them moved more than 60 nautical miles over that entire period. Jeff Kneebone has also been doing extensive amount of tagging, and he's told me that the majority of the animals that he's tagged in the Gulf of Maine move less than 30 nautical miles. And Jeff was the person that originally motivated us to become involved in the project, as I mentioned right at the beginning. So the expectation, if these animals don't move much and they're spread all throughout the North Atlantic, is that there would be individual discrete populations that have got their own genotypes that would be recognizable in different regions. And this has motivated Arve Lingerhammer, one of our collaborators, to look at this. And so Arve used molecular analyses. He got about 80 samples from the Northeast Atlantic, from the eastern coast of Greenland, and from the West Atlantic, and he also sampled both of these size classes. And what was peculiar and surprising to Arve is he didn't see any genetic differences that allowed him to distinguish between the two size classes in the Northwest Atlantic, and there didn't appear to be any significant geographic structuring of the data. This was somewhat counterintuitive. It was not what was expected. And so we're presented with this conservation conundrum. The population hasn't recovered in the Northwest Atlantic, in the Gulf of Maine, despite 14 years of protected management. And this is in stark contrast to the other skate species in the Northwest Atlantic, specifically the barnbill skate, which is doing reasonably well. By contrast, on the other side of the Atlantic, the Northeast Atlantic biomass seems to be stable. There is some suggestion very recently that this is changing a little bit, but over the period that we've looked at, it seems to be pretty stable. The tagging suggests that we should expect regional clustering, but the molecular data doesn't support this, at least the molecular data that's been collected so far, largely by Arve. And in 2011, there was a petition for ESA status, but NOAA determined that a status workshop wasn't actually warranted. There was a new petition by the Defenders of Wildlife and Animal Welfare Institute in 2015 to protect the northwestern population. And in 2016, there was an extinction risk workshop that actually determined that there was no distinct population segments that could be identified, and therefore no risk of extinction throughout or in any significant portion of the range. So what we have here is a real-time conservation debate that's associated specifically with the determination of stock structure. So this is where we decided that we're going to have a look at this using very extensive sampling and genomic approaches. And so now I'm going to pass the mic over to Shannon Corrigan, who's going to talk a little bit about some background about the approaches that we can use to assess stock structure and some of the limitations and strengths of different approaches.
Okay, thanks, Gavin. Hi, everyone. So I just want to start off, first of all, defining what we mean by populations versus stocks in relation to conservation management. So a fish population is a biological unit referring to individuals of a species living in the same area. When we refer to a fish stock, what we're referring to is a management unit, and these are individuals that are grouped by similar demographic or genetic characteristics that we think indicate that that group will respond uniquely and independently to fishing pressure and therefore require independent management. So there's a number of different ways to define stocks. You can look at shape or count variation in a morphological trait. You can look at physical movement between locations or lack thereof, which you can determine using tagging or tracking data. Otolith microchemistry, fatty acid profiling, and stable isotope analysis are often used as tools for identifying environmental differences among individuals or dietary differences that might indicate stock structure. And then finally, genetic markers are very commonly used to examine patterns of population genetic structure to inform when there may be independent stocks that require independent management. And this is what we're going to be talking about in more detail today. So over the last couple of decades, there's been a gradual transition from population genetics approaches to more large-scale population genomic studies. And this has happened as DNA sequencing technology has become ever more high throughput. So today, often earlier population genetic studies are referred to as having used traditional or legacy population genetic markers. And these typically include studies that have relied upon analyzing one or a small number of genes or even gene fragments from throughout the genome. So for example, previous studies of the Thorny skate have analyzed fragments of the mitochondrial cytochrome B gene and the cytochrome oxidase 1 genes from the mitochondrial genome. And similarly, other studies that you may have seen in the literature have focused on one or a small number of fragments sampled from throughout the entire nuclear genome. So some traditional legacy genetic markers you might be familiar with are the Barcode of Life initiative or fragment-based approaches such as nuclear microsatellite analyses. So the idea here is that we're using surveys of these small sections of the genome to approximate variation at the higher level. So we assume that variation that we see in whatever fragment we're interested in looking at is going to be indicative of variation that we would expect to see at the level of the whole gene, and that this reflects processes and variation at the genome level, and then we use these patterns of variation to make inferences about organismal biology. So these approximation approaches have been very widely applied with good reason. This is because they've been largely cost-effective and they've been really very informative. However, because these approaches can also be somewhat limited in the amount of information that they can convey, and this is simply because every gene in the genome evolved along its own independent evolutionary trajectory. And so each gene can potentially tell a different story, as depicted here in this slide by a phylogenetic or population gene tree, as inferred from 10 different genes, each of which is just colored by a different color. So you can imagine here if we were to use each of these 10 genes to infer a parameter of interest, just for example's sake, we're going to think about the time to most recent common ancestor, which we've highlighted in this blue circle. You can see that we'd observe quite some variance in our estimates of this parameter simply because of the different gene histories. So different genes can suggest different inferences, and this can lead you to arrive at different conclusions if you're only sampling a subset of the genome. Likewise, more information from a single gene, or if you have a subset of fragments that happen to be biased, you can converge on the wrong conclusion, but with confidence. And so the challenge as we move into the genomics era is making sense of all of this noise in such a way that we can make accurate biologically meaningful inferences while still making the best use of the data that we possibly can. So to put this in the context of the Thorny skate and previous work that has been done, so a previous study by Chivolo et al. Inferred a recent population expansion in Thorny skate, and this is somewhat at odds with the inference of potentially declining population sizes as indicated by the declines in landings that we've observed. However, it's important to keep in mind when we are interpreting this result that although the signal was strong in this study, it was based on just a short fragment of a single gene from the mitochondrial genome, in this case cytochrome B, and spatial sampling was largely focused around Iceland. So it doesn't really give us a clear picture of population dynamics across a species range or genetic structure across a species range. Similarly, the most recent work by Linghammer et al., which was really the precursor to this study, and was based on a much larger number of nuclear microsatellite markers. So now we've moved to the nuclear genome, increased the number of independent gene regions that we're sampling to 10. And it has really great spatial sampling, which we're now able to take advantage of in the present study. And in this, interestingly, in this work, he was able to show that there was some evidence of some population structuring throughout the range, where the nuclear genome was showing that we could split the species range into 3 genetic clusters: the Northwestern Atlantic, Greenland, and the Northeastern Atlantic. However, these differences were really only very subtle, and they provided a different pattern than what was observed in the mitochondrial genome, as Gavin mentioned earlier. And so this suggests that perhaps that even these 10 loci are not providing a sufficient resolution to really get a full understanding of what's happening in terms of Thorny skate population genetic structure. Okay, so don't be scared by this slide. It's pretty busy, but I'm going to walk you through it. So these gene tree effects that I've been referring to, they're being increasingly realized and assessed through the use of larger scale sequencing studies that are now available in this genomics revolution. And this has led to the development of genome-wide sampling techniques. And I mention this because you're probably familiar with things like RAD-seq or target gene capture that you've seen in the literature. So this is just comparing each of those two methods. But here the takeaway message is the goal of these methods is to simultaneously survey a very large number of genetic markers from throughout the entire genome of an organism, rather than focusing on just one or a small number. So in target capture, the entire genome is randomly fragmented, and then you target particular regions that you deemed that you're interested in with a probe that's designed a priori. In RAD-seq, we fragment the genome at particular restriction sites. These are indicated in these little black bars here. And then you sequence and align those particular fragments. So these approaches are excellent for surveying a large number of independent genomic markers. However, because we don't usually have a complete reference genome sequence that gets switched to align all of these data, it can be hard to ensure orthology of these fragments across individuals. So to know that we're comparing the same fragment in one individual as an identical fragment in another individual, population or species, and then also it makes it hard to ensure reproducibility among studies and comparing marker data sets among studies when you don't have this reference genome to refer back to. So we wanted to take all of this one step further in the Thorny skate project. By beginning with a complete sequence of the Thorny skate genome. And John's going to talk a little bit more about this coming up. And then from starting with this complete sequence of the Thorny skate genome, we want to take a genome-informed marker design approach. So we're going to start with a complete genome sequence and filter this to generate comprehensive subsets of the genome that we think might be of interest for addressing particular questions that we're particularly interested in about the Thorny skate. So we might decide that one subset is particularly useful for assessing population genetic structure and stock structure, while a different subset might be useful for looking at adaptive divergences over time or changes in population demography and population size over time. So the other really nice thing about adapting a genomic approach and a genome-informed approach to marker design is that it builds a lasting genomic resource that becomes the absolute standard for the studies of whatever organism you're looking at. So because the complete genome is the absolute standard, the previous reference will always be clear from this point forward. So even as technology changes, we'll always have this reference to refer back to and to link all of future data sets back to. So it should reduce the chance of issues with marker reproducibility or non-orthology across studies. And because everything is standardized against the complete genome, effectively any prior study that has been done on the organism can be also added to your reference pile of information. So just to summarize why we want to adopt a genomic approach in this, there is some overlap in the kinds of questions that you can address using traditional genetics versus genomics methods. But generally speaking, as you increase the number of markers surveyed from the tens to 100s that is typically used in a genetics approach into the thousands, for a genomics approach, your resolution generally increases and you start to see less chance of misleading inference due to that gene tree heterogeneity that I was referring to earlier. So adopting a genome-informed genome-wide sampling approach that we're proposing also allows you to design smaller, cost-effective marker panels that you can tailor to address specific questions of interest. And then finally, in doing this, you're also building a lasting genomic resource for an organism that can then be interrogated long into the future for addressing questions. And there are some questions that simply can't be addressed with a genetics approach and areas of research that you may be able to move into using a genomics approach. So things like looking at genomic selection, gene expression, and epigenetic responses. So by building this genomic resource, we have lots of room for growth and a lasting resource that will be used for us to reinterrogate as new questions as they arise. So now I'm going to hand over to John, and he's going to be discussing our experience of sequencing the Thorny skate genome and present some of our empirical results for the project thus far.
Cool. Well, thanks everybody for coming this morning. Really fortunate to be involved in this project. I was brought on board to work on pipelines and data analysis methods to best realize these goals as both Gavin and Shannon have outlined them. And one of the things that is really hot off the presses here is some of this sequencing that is being done for the Thorny full nuclear genome. It's being done in collaboration with the Vertebrate Genomes Project, which is a colloquium of leaders in the field, basically, and using the most up-to-date sample preparation method, sequencing methods and protocols, and assembly protocols. And the goal is to establish uniform, universal guidelines for what are being referred to as platinum quality genomes that will serve as future universal reference points for data sets and analyses at the genomic scale, universal reference points in the sense as Shannon mentioned. It's worth noting the Thorny skate is among the first set of the genomes the VGP has been working on. And also that the Thorny skate genome analysis and assembly has inspired modifications to previous versions of the VGP assembly pipeline. And that pipeline is being used to assemble all of the VGP vertebrate genomes. So in that sense, the Thorny skate is already contributing to bioinformatics more broadly. The VGP has several quality metrics they use to define platinum quality genomes. There are 4 general metrics, the first 3 of which have to do with differing hierarchical levels of assembly completeness and contiguity, and the last one has to do with individual nucleotide call quality. As of July, the Thorny skate genome has satisfied 2 of these assembly contiguity requirements. I am told that slightly more recently, the base call quality is up to QV40. So we have right now about 3 of these 4 requirements met, and that the assembly is currently being sent out for chromosome level assembly. Assembly is an iterative process, and so multiple drafts are needed. It's also worth noting that the genome size, based on the assembly output so far, is estimated to be about 3.8 gigabases, which is approximately equal to an independent estimate produced by a method via flow cytometry. So that's pretty encouraging. So the overall scope of this project is to take comprehensive specimen sampling from across the geographic range of the Thorny skate, sequence complete mitochondrial genome information, and also, based on the nuclear genome that we have been talking about so far in this webinar, put together genome-informed targeted nuclear markers like Shannon mentioned, and then use analytical tools that can leverage data at this spatial resolution and at this genomic scale, with the goal of providing data that can be used to generate conclusive assessment of distinct population segments, as well as infer temporal dynamics in demographic and ecological parameters of interest. So I'll be talking a little bit about some preliminary results that we have, but overall, the geographic sampling that we have put together here is represented by the little red dots inside the overall green polygon, which represents the full range of the Thorny skate. And you can see several very encouraging things here, right? We have plenty of Gulf of Maine samples, thanks in large part to Jeff Kneebone. And we also have lots of samples from Iceland, thanks to Clara Jakobsdottir. So that's really good. We're working on assembling those right now. So today, what I'll be talking to you about is 335 samples from 6 regions that were selected to broadly resemble those of the studies that were conducted by ARVE. Again, we are adopting a targeted next-gen approach. We've spoken about the outline for what we're going to be using the nuclear genome to do. But what I'll be talking to you about right now is some mitogenome-based results, and that's the upper arrow that you see here in this figure. Right. So why redo the mitochondrial data? It's only a single locus, and we have some of this information already. And we've seen in earlier slides that we have some inferences generated from the mitochondrial genome. The full genome is 16,000 and change base pairs. But as you will recall from previous slides, we know that different subsets of a single locus can potentially give different signals. And so if we sequence the full mitogenome, we will know for sure we have the complete representation of the signal from this locus to compare with nuclear data once it becomes available, because we are sequencing both genome types from the same sample sets. So if structuring is inferred from the complete mitogenomic data, it can potentially be used to inform sample selection for assembly of other geographic samples against this full annotated nuclear genome. And just as a way of summary about the mitogenome's quality that we have produced so far from these 335 samples, the average coverage depth for each of these mitogenomes is about 400x. So they are pretty well covered. And the number of ambiguities over the complete mitogenomes is about 4 base pairs or fewer. So they are good coverage and low ambiguity. So as a graphical summary here, what we have is a haplotype network for these 335 samples. Each circle indicates a unique haplotype with size of each circle proportional to the frequency of that haplotype. And we see that nearly every individual has a unique haplotype, except, there's lots of variation, but no geographic structure, right? Because these circles are color-coded by geographic region, with the exception of what you see here highlighted in blue. That is a subset of the Newfoundland, Labrador, and the Gulf of Maine, New England haplotypes, which appear distinct. However, when we add in size class as an additional coloration to these points, we see that those 2 different shades of green here are distributed a little bit outside this cluster on the right, in a way that suggests that size class is not structuring these haplotypes, a result that is consistent with Arve's previous work. When we conduct an analysis of molecular variation to decompose the genetic variation by some categories of choice, here we tested a west versus east stock structure. So the blue line here indicates where that west versus east demarcation was decided upon, and put the 6 populations that you saw from the previous slides in here as well. So we have a 3x3 design set up. We see that most of the molecular variation is explained by variation within groups, and this east-west structuring is not significant. When we go a little bit further in and look at complete pairwise comparisons of differentiation among all of these different geographic regions, we see that the Gulf of Maine and New England samples are still distinct in pairwise comparisons, and also that some of the Newfoundland Labrador samples are distinct from all of these others as well. So these Gulf of Maine, New England, and Newfoundland and Labrador samples appear to be what is driving the significant AMOVA FSC and FST results. When we conduct a regression of the pairwise geographic distance among populations on a linearized measure of genetic differentiation, we have inferred a significant relationship as well. So there is a significant isolation by distance here inferred from the complete mitogenomic information of these 335 samples. So to conclude here, we are inferring a distinct subset of the Gulf of Maine and Newfoundland Labrador samples, but this distinctness is not apparently linked to size morph, and so the actual explanatory factor here is still undetermined, but it is a compelling preliminary result. We have the other group distributions do, however, appear to agree with previous mitochondrial data-based results that suggested no geographic partitioning. So overall, this is a very busy summary slide, but the take-home message here that I hope you get from the graphic is that this is starting to become a very active area and project with lots of inputs from different agencies and researchers, and a lot of collaborative effort brought to bear to solve this problem. We are very close on our genomic data and on the complete assembly of the nuclear genome. Once we have that in hand, how we proceed with target bait design will be determined somewhat by the patterns we see in the complete annotation. So we're on pins and needles, and we're really excited about that. We do plan to sequence from the same libraries for the nuclear information that we have sequenced the mitogenomes for, so we have paired information and we can compare these things. And so we anticipate more results, more independent loci, and more outputs expected by the end of next year. So please, please stay tuned and keep in touch. We'd, of course, like to acknowledge many, many collaborators, especially the LenFest Ocean Program, the University of Florida and the Florida Museum of Natural History, Rockefeller University, the Genome 10K Initiative, and the VGP, especially Olivier, as Gavin mentioned, but also Arang Ree and Adam Philippi, two of the bioinformaticians who have put their heads together to adapt the assembly pipeline for the Thorny Scape nuclear genome assembly. Jeff, of course, for introducing and motivating the problem. Arve for being so generous with sample access. Rupert Weinfrother, Dave McElroy, Clara Jacobsdottir, Mark Simpson for also providing advice samples and just being wonderful examples of collaborative research. And also, we'd like to thank Lindsay Gutteridge for the skate illustrations. And with that, I think we'd be happy to take questions.
Great. All right. Well, thanks so much to all of you for that great presentation, outlining the work and where you guys are thus far. Just a reminder to folks who are watching, you can submit questions by typing them into the Q&A panel on WebEx. So feel free to type those in. But I'd be happy to kick stuff off if that's all right. It was great to get a sense of some of the initial results. And I was wondering, certainly you spoke about those two different size morphs in the Gulf of Maine, and it strikes me that's a pretty surprising finding that there's not any differentiation there. I was wondering, do you guys have any kind of hypotheses as to why those two morphs might exist, and why and not have any genetic or genomic differentiation?
I think so. I'll start, and maybe Shannon or John can follow up. But it's very likely these animals are sympatric. If they are in fact genetically distinct, it's possible that there's leakage and hybridization, and you only need a little bit of hybridization, and so some of the mitochondrial elements could have been sort of contaminating these from one form to another, and that would explain the heterogeneity in distribution by size class. If that's the case, then when we start looking at the nuclear genome, we should start seeing differentiation in the nuclear data that is not revealed by the mitochondrial data.
Got it. That's helpful. So in other words, that result is sort of a work in progress. There might be more coming, depending on the outcome.
So can I just add a little more to that story?
Yeah,
sorry to interrupt you. So this one might be potentially a little bit more speculative, but we're thinking that if there are the size morphs are indeed structured and they're sharing mitochondrial DNA, then that would be a case of mitochondrial hybridization. So we're thinking that that's one possibility. But another possibility, if that doesn't turn out to be the case, is that you can actually get quite drastic differences in phenotype that are under epigenetic control rather than genomic control. So your epigenome changes in DNA methylation and that result in rapid changes in gene expression and can induce a change in basically morphology really quite rapidly without an underlying change in your genomic sequence. So this could be a case of something like that that has been demonstrated before in a separate skate species in response to subtle differences in sea surface temperature. Leading to changes in growth rate and things like that. We don't know if that's the case here. As far as I'm aware, these two size morphs appear to be completely sympatric in some areas, even though there is an overall gradient of size classes. So that's just one other possibility that we have in our mind that would be really very interesting to explore if it does turn out that there doesn't seem to be any genomic differences between the size classes.
Absolutely.
Great, thanks. And one other thing, just before we keep going, in case some more questions trickle in, Shannon, would you mind passing presenter privileges back in this direction, and I can put up your contact information on the slide so folks can get in touch with you if any ideas strike them after the call here.
I promised I would do that without muting and unmuting, but I failed miserably.
Not a worry.
Okay, I'm going to pass the privileges back.
Okay, perfect. Great.
I think I was successful.
Yep, you were indeed. Stand by here. Okay, great. All right, we've had a couple of other questions come in. Let's see. With your initial results that the Gulf of Maine skates are separate, are there genomics applications that could potentially tease out the mechanisms that have changed population size of those separate populations? And I assume that's referring again to those separate size morphs that you were alluding to in the previous question.
Yeah, that's a really great question, and that's precisely some of the nuts and bolts that we're going to try to get into once we have the complete genome sequence finalized for the Thorny skate, and then we can look at different regions that are going to be very interesting to target to address some of those questions. So we're very interested in like historical demographic processes, the changes in population size over time. So we'll be looking at that very closely using the genome-wide nuclear data. One possibility is that perhaps that Gulf of Maine area was a refugium, and then they've recolonized the rest of the range from there, and so we see increased levels of variation from that area. But these are the kinds of questions that we're going to try to explore. We don't know exactly what's going to be of interest until we can look at the complete genome sequence, but we're also, obviously, we can look at signatures of selection, and things like that, comparing the Gulf of Maine region to the rest of the distribution to start to get an idea of like what might be driving those differences. So unfortunately, it's a very intriguing pattern to me right now to see so much diversity that's only found in those two locations, and even that there is some subtle differences between Newfoundland and the Gulf of Maine. So that's something I'm very interested in exploring in terms of the mechanisms that are driving that when we get into the full genome project.
Great, and that's actually a great segue into the next question or comment here, which is, I guess, collaboration in action, but it's from Carolyn Meary at DFO in Newfoundland, and wants to make sure that you have enough Thorny skate samples from Newfoundland waters. And she's mentioning that, like in the Gulf of Maine, they also have two size morphs, and want to make sure that you have samples from both of those size morphs up in Newfoundland.
Interesting.
Great.
So I don't think we have got, we may unwittingly have both size classes from Newfoundland, but we weren't aware specifically. John, do you know if we have classifications of which size morph they are purportedly from? We have some from Newfoundland, but...
Yeah, we're putting together a preliminary database of the phenotypic information that we have metadata for all of our samples. I don't remember off the top of my head whether or not we had a non-GAPI section of that information for the non-Gulf of Maine New England samples, but I can definitely check on that, and would be happy to keep in touch by email on this.
Great. All right. So our next question kind of moves out of the research and more into the management space, and perhaps something that we at LenFest can speak to as well. The question is, can you talk about your plans to deliver results to fishery managers, such as, which managers, through what meetings, and how often, and how you might anticipate having some of your findings translated into management measures?
So I guess I'll take that. So absolutely. So the first thing that we need to do is to characterize the variation. And if we find that there are distinct haplotypes that are regionally restricted, then that needs to be translated into policy whereby takes are defined by those restricted haplotypes. And until we have these data, we can't counsel people as to how to proceed, and so it's going to be completely empirically based. How policy and management actually implement actions that are based on the patterns that we see is something that I'm not familiar with, but we can provide the raw material, and we can say there's a haplotype that's distinct here. These need to be treated as an independent stock. They're not moving, or we might find a different haplotype that is an independent stock, but they're migrating seasonally, and we can probably identify corridors of movements of particular haplotypes, both in contemporaneous populations and, if we have enough resolving power, in historical samples. So we can deduce historical patterns of movement. And this can be incorporated by managers, and some of it will not be practical to deploy, and other parts will be practical. And so these are, we're, as scientists, we are happy to make the data available, and but we don't know enough about the practicalities of fishing gear and seasonality and economics to be able to guide people. We hope policy people will take what we provide and make actionable policy that can be derived from it.
Great. Thanks, Gavin, for that explanation. And I want to emphasize that we here at LenFest will be working with Gavin, Shannon, and John too as their project evolves to basically to help disseminate those results. And if anybody on the call has recommendations for places to help deliver these results or folks to engage, please reach out to us at LenFest as well as to the research team, and we'd love to hear your input and help to get this work into the hands of folks who can use it moving forward. So our next question comes back to that notion, Gavin, that you talked about with the multi-species skate complex in the Northeast, and the question is, have you thought about looking at winter or little skate, which are cryptic species, to test for size morphs?
Yeah, that's a good question. We haven't. It turns out that in elasmobranchs, there's a lot of cryptic species. And the more you look, the more you find. And it's actually a very good point. There are these studies that have been done in the last few years that actually suggest that the gene expression patterns in some of these forms are distinct and different. And so it's a bit of a conundrum whether or not these are just ecophenotypic variants that look different because they've got different gene expression profiles, or if they are distinct species, cryptic species. And for example, there's not just in skates and rays, but in sharks, we have sharks in the genus Squalus that are morphologically very, very different. And the fishermen can very, very easily put them into different piles. And we cannot tell the difference genetically between these things. So we suspect that these are an example of epigenetically regulated animals, that their environment induces different patterns of gene expression, which then results in different morphologies. And so it's an excellent question, and it's one that we will probably branch into in the future, but we haven't considered doing that yet. We wanted to see if we could sort out the thorny skate first, and then maybe see if the findings from the thorny skate study can be applied, or if they reverberate to other skate populations in the area.
Great. Thanks, Gavin, for that explanation and clarification there. So moving along, I believe that the New England Fishery Management Council is having an annual meeting soon, and its agenda includes a discussion of the Northeast Skate Complex. And the question is, will you be presenting this information to the council at the upcoming meeting?
We can certainly be happy to present the information if that would be helpful. No problem at all to do that. Right now, we're sort of a work in progress. We haven't got the full genomic data, which will likely change the recommendations and the inferences. We're putting a lot of stock in having a super high resolution genome and tens of thousands of markers that will allow us to look at demographic history and gene flow. We do not have these data yet, so it might be a bit premature, but if the council feels that it would help them, then we'd be happy to take a trip up and present the progress so far.
Great, thanks. And just to follow up on the earlier question about getting this into the hands of managers, the question is, your fact sheet suggests certain results might trigger reevaluation under the US ESA, but such a process is not automatic. And do you have a plan for prompting review if your results show evidence of a US distinct population segment? And I'll just jump in there first to say that generally, from a LenFest perspective, our projects are meant to sort of, the projects that we support, that Gavin, Shannon, and John are conducting is really meant to kind of to get the word out there about the results and to basically demonstrate that there might be, that there could be a need or there could not be a need to look at questions like this. But in terms of kind of weighing in as to whether or not they merit an ESA review, that probably is outside the scope of this. But certainly, the results could inform action by others to do something like that. And Gavin or others, I'm not sure if you have anything else you'd like to add on that.
No, I completely endorse what you said. We're going to get data patterns and we will report them and make them available, but we don't really give guidance on these issues. Many attributes from conservation biology, policy, managers, economics feed into these things, and so we just provide the scientific data in an unvarnished way for other people to use as they see fit. So we're happy to do what we do reasonably well and make it available to others who use those data in ways that we are less experienced with.
Great, thanks. And just actually relevant to that, Toby, I hope you don't mind, but Toby Curtis from NOAA Fisheries commented that he's happy to help introduce these results into the relevant U.S. Fisheries Management Fora as well.
That'd be great. Hi, Toby.
Hi, Toby.
Hi.
So let's see. Another item related to the New England Council is just an FYI that the council has at least one skate committee meeting coming up in the coming weeks or months, and that attending would be a great way for you guys to learn more about the process that you would like to inform.
Absolutely.
Yep. And let's see. So another question from Carolyn up at DFO. As she suggested for Thorny, she wrote smooth skates, but I think she meant thorny skates. We in Newfoundland have a species ID conundrum between Arctic skates and Jensen skates. And the question is, can your amazing genomic techniques be applied to both of these species in order to differentiate their quote-unquote external morphometric type similarity?
Well, first of all, I'd like to acknowledge that Carolyn was the person we first communicated with, and she sent us the sample, so thanks, Carolyn.
Yes.
And absolutely, if there are genomic differences that underlie these two forms, then we should be able to detect them. It's quite possible, though, as in the case of some of these other peculiar Australian squalus, that we might find that genomically they are very, very similar, and yet it might be a gene regulatory difference that's responsible for different behaviors or morphologies if they're detectable. So But we can certainly apply these methods, and if there are genetic differences there, we would find them, absolutely.
Great. Thanks for the clarification there, Gavin. Just one more very useful piece of input here, and certainly we're keeping track of all these, and we'll send these comments along to you guys as well. That NAFO, the North Atlantic Fisheries Organization, will be updating its Thorny skate assessment in June of 2020. So that is something also to keep in mind as reviewing management measures for the species will be on the table in September of 2020. So that's certainly something that us here at LenFest and that you guys should be aware of. So thanks for including that as well. So
Absolutely.
we just got one. Yeah. So we've just got one question left, and I think it's a good question to sort of funnel us back out to conclude the webinar. And the question is, you mentioned that this genomics-focused project could be applied to other species of sharks or bony fishes that are managed. And I guess the question is, how was the project designed so that it could potentially be applied for other species in other management contexts?
Oh.
Yeah, go ahead.
So I guess technically the Thorny skate whole genome will be potentially useful for closely related species, but when we were saying that, I guess we were more talking about our overall pipeline. And so our goal at this research was to build on the previous work where we were looking at the small number of markers and then working with the VGP to get access to the complete genome sequence or to create the complete genome sequence, and then work backwards so that we're using this genome-informed marker design approach. And so I guess we're saying that that kind of approach is something that would be applicable to other species. So we're hoping that if it ends up that this provides really useful information for clarifying some of these issues with the Thorny skate, that people will use it as an exemplar for how these kinds of genomic technologies can be applied to other species.
Yeah, sorry.
You can go ahead.
Okay. So structurally, we could take a similar general trajectory of getting a high-quality nuclear genome, good annotation, good resolution for another species of interest, and then take a similar pipeline sequencing sampling approach, but then also take the genomic subsets based on that genome for that species applied to whatever questions of interest apply specifically to that species. So it's basically you take out the elements of the Thorny skate and replace it with the species that you'd be interested in working on.
And so just add one more thing to that. So if you're someone who's interested in studying different species and you don't have funding or you don't have access to whole genome resources, you might be thinking, well, this isn't applicable to me, but that's where I would point people toward the VGP. And just there are a number of genomic resources that are coming online is just ever increasing and will continue to do so as we move forward into the future. And so it's really worth keeping tabs on that. The nice thing about the VGP work is that it's a dedicated effort to try and collect whole genome sequences from across the tree of life. And the most important thing is that they're really focusing on the quality and utility of these whole genome sequences. And so as we move forward into the future, we're going to enter an era where we're really able to do some nice comparative genomics work using these kinds of resources. So even if a complete genome sequence isn't available for your species of interest and you're unable to generate one, first I would encourage you to try to work with them to do so, but if that's not possible, hopefully something that's closely related enough will be available for you at some point in the future and be able to be used.
And the other thing is that from a fisheries perspective, what we're doing here is not so different from what has been done with human genetics. People have looked at historical demography, migration patterns of humans around the world, out of Africa, across Europe. And so we're doing very similar things, but at the genomic scale, but we're doing it in commercially important marine fishes. And so we hope that people will turn to the Thorny skate example and say, these people have done this with a Thorny skate. We can do this with a number of different species. And as Shannon said, it may not be necessary to get the high-quality reference genome. You might be able to use one that's close enough to be able to do the same kinds of, pursue the same kinds of questions. But we're optimistic that this will sort of put a sort of line in the sand, and people say, well, we can use these techniques to better manage marine resources of organisms that are spread, oceanically, from all the way across from northern Canada, across Greenland, Iceland, and up to the Arctic Ocean. Even though these animals are widely spread, they may show discrete population segments which need to be managed accordingly. And so we're hopeful that this might inspire others to do similar things with genomic kinds of data.
Great. Well, thank you all for your input. I think that's a fantastic note on which to end the webinar. It is 1 minute after the top of the hour, so I think we can go ahead and wrap things up. Again, thanks so much to Gavin, Shannon, and John for giving us an update on their project on Thorny skate genomics. If you do have any further questions about the project or suggestions for collaborations or for which to share some results, feel free to email the research team or me, Willie Goldsmith, here at LenFest. And we look forward to keeping in touch with you. And as I mentioned, we'll have a recording of the webinar ready to go in the next few days here. So thanks so much, everybody. We're going to close out the webinar now.
Thank you very much.
Thank you so much.
Thanks.